Every time one of your cell divides, it has to copy roughly 3 billion base pairs of DNA. And it does this with an error rate of about 1 mistakes in a billion nucleotides. That is not luck, that is just chemistry, which is executed by a team of highly specialized enzymes working together at a moving structure called the replication fork. In this video, we are going to open up that box. We'll look at every enzyme involved in DNA replication, and we will understand not just what they do, but why the chemistry forces them to work the way they do. So let's get into it. Before we name individual enzymes, hold this picture in your head. DNA replication is semi-conservative. Each old strand serves as a template for a new one. To copy it, the cell assembles a molecular machine called the replisome at the replication fork. The fork is simply the Y-shaped junction where double-stranded DNA is being pried apart into two single strands. Ahead of the fork lies untouched paternal duplex. Behind it lies two newly separating template strands. Every enzyme we discuss today has one job at this fork. Open the helix is done by helicase. Keep it open is done by single-stranded binding protein. Relieve the strain that opening it creates is done by topoisomerases or gyrases. Start a new chain is done by primase, and build the new chain accurately is executed by DNA polymerases. Keep that logical order in mind. The enzymes appear in exactly that sequence at the fork. Let's talk about helicase, the unwinding engine. The first enzyme to act is helicase. Its job is to unwind the double-stranded DNA by breaking the hydrogen bonds between complementary base pairs of the two strands. Notice the word choice. It breaks hydrogen bond, not the covalent phosphodiester backbone. So helicase does not cut the DNA. It just separates the two strands like opening a zipper. Helicase belong to AAA plus ATPase superfamily. That ATPase is associated with diverse cellular activities. And as the name tells you, it requires ATP to function. The energy for unwinding comes from ATP hydrolysis. ATP is broken down to ADP plus inorganic phosphate, releasing energy. Every base pair the helicase breaks costs the cell chemical energy. Unwinding is not spontaneous. The replicative helicase is a hexameric ring. 6 subunits arranged in a donut shape that encircles one of the DNA strands. In E. coli, this ring is protein DNA B. In eukaryotes, the equivalent is MCM2-7 complex. As the ring is threaded onto a single strand and pulled through, it physically forces the two strands apart. Helicases are directional. They have polarity, either 5' to 3' or 3' to 5', meaning they can only translocate along a strand in one defined direction. This matters because the two template strands are anti-parallel, and that anti-parallel geometry is the whole reason replication looks asymmetric. Here's how that plays out. On the leading strand template, the polymerases can follow the fork continuously, synthesizing one long unbroken new strand. New DNA is read off a template running in 3' to 5' direction relative to the fork movement. On the lagging strand template, the geometry is inverted, that is 5' to 3'. So, the new strand can only be made in a short pieces, which is discontinuous synthesis, producing the famous Okazaki fragment. So one enzyme, helicase, opens the fork, but because DNA is anti-parallel and polymerases have a fixed direction, that single event forces the leading strand to be built smoothly and the lagging strand to be built in chunks. Hold that thought. It will matter more when we reach the polymerases. Next up, we will talk about single-stranded binding protein or SSBP, which keeps the fork open. The moment helicase separates the strand, we have a problem. Single-stranded DNA is unstable. Those exposed base want to repair. Left alone, the two strands would simply snap back together, or a single strand would fold back on itself into hairpins. Let's define the fork precisely here. The DNA replication fork is the junction between the newly separated single-stranded DNA and the still unreplicated double-stranded DNA. Single-stranded binding protein binds along these freshly exposed single strands. Their functions are they prevent renaturation. That is, they stop the complementary strands from reforming hydrogen bonds and snipping back together. They stabilize the single-stranded DNA and keep it extended and accessible. They protect the exposed strand from being chewed up by nucleases and forming secondary structures. Two important key properties for your exam to remember. They are template independent. They don't care about the base sequences. They'll coat any single-stranded DNA. They bind with cooperativity. The binding of one single-stranded molecules make it easier for the next one to bind right besides it. So they coat the strands rapidly and in coordinated way, one after another, rather than landing randomly and slowly. That was all for single-stranded binding protein. Next up, we will talk about topoisomerases and DNA Gyrase, which relieves the strand. Now think mechanically. If you take a twisted rope and start pulling it apart at one end, the twisting piles up ahead of what you are separating it from. DNA is exactly the same. As helicase unwinds the fork, it generates torsion stress, positive supercoiling ahead of the fork. So, unreviled, this unwinding would eventually stop the fork dead. The enzyme that solves this problem is topoisomerases. What are they mechanistically? Topoisomerases are nucleases, enzymes that breaks the phosphodiester bond of the DNA backbone. They allow the DNA to shrivel or pass through and then reseal that same bond. Because they can cut and rejoin the backbone, they are introduced or removed supercoiling. They change the topology of the DNA without changing its sequence, hence the name topoisomerases. So topoisomerase 1 makes a single strand nick. It just cut one strand of the DNA and this lets the DNA rotate to relax the supercoil. Topoisomerase 2 makes a transient DNA stranded cut. A temporary break in both strands passes another segment of duplex through the gap and reseals it. These steps require ATP. The classical prokaryotic example of topoisomerase 2 is DNA Gyrase, and it has a special talent. It can actively introduce negative supercoils into the DNA, which is exactly what helps the fork stay open. So Gyrase subunits are Gyrase A, which carries the catalytic site. It is responsible for breaking the phosphodiester bond. Gyrase B carries the ATP binding site, which provides the energy. Putting the mechanism together, Gyrase A breaks the phosphodiester bond, releases the positive supercoiling and introduces negative supercoiling, an unwinding anti-clockwise twist that keeps the strands relaxed and ready for replication. Gyrase B fuels this using ATP. Next, we are going to talk about Primase. DNA polymerase is the enzyme that actually binds the new DNA strand, cannot start a chain from scratch. It cannot lay down the very first nucleotide on a bare template. It can only extend a chain that already has a free 3' OH group to add onto it. De novo literally means from the beginning, and DNA polymerases can't do that. So the cell needs something to lay down that first short starter piece. That something is primase. What is primase? Primase is a RNA polymerase enzyme, specifically a DNA dependent RNA polymerase that synthesizes an RNA primer. In E. coli, this example is called DNA G. What is primer? A primer is a short nucleotide sequence about 8 to 12 nucleotide long, built from ribonucleotides, RNTPs. The same building blocks as RNA. Because it's made of RNA, primase can start from scratch. RNA polymerases don't need a pre-existing 3' OH. Once primase lays down this short RNA primer, it provides the crucial 3' OH end that DNA polymerases has been waiting for. Now DNA polymerases can step in and start extending. The golden rule, 5' to 3'. Before we meet the polymerases, one rule governs all of them. DNA synthesis always proceeds in 5' to 3' direction. New nucleotides are always added to growing 3' OH. The phosphodiester bond that links each nucleotide forms as the incoming nucleotide. 5' phosphate joins the chain. So the bond formation is read from the 3' to 5' side of the chemistry. Mechanistically, the 3' OH of the last nucleotide attacks the innermost phosphate of the incoming DNTP, forming the phosphodiester bond and releasing pyrophosphate. The hydrolysis of that phosphate is what makes the reaction energetically favorable and effectively irreversible. This one direction constraint, 5' to 3' only, is the reason lagging strand has to be made in Okazaki fragments. The polymerases simply cannot run backwards. Now let's talk about prokaryotic DNA polymerases. E. coli have 5 DNA polymerases. Let's learn about them and their job. DNA polymerase 1 is involved in DNA repair and polymerization. Crucially, it removes the RNA primer and fills the gap. RNA polymerase 2, an alternative repair polymerase, also helps restart staller replication forks. DNA polymerase 3, the main DNA replication enzyme. This is the workhorse that does the bulk of new strand synthesis. DNA polymerase 4 and DNA polymerase 5. These belong to Y family of DNA polymerases. They lack proofreading activity and are involved in DNA repair, specifically translesion synthesis. Copying paste damages bases during the SOS stress response. Because they don't proofread, they are error prone. Now let's zoom into the two most important polymerases, DNA polymerase 1 and DNA polymerase 3. DNA polymerase 1, the unusual enzyme. DNA polymerase 1 is nicknamed as the unusual enzyme because it carries 3 distinct enzymatic activities in one polypeptide. 5' to 3' polymerase activity. It builds DNA. 3' to 5' exonuclease activity. This is proofreading. If a wrong base is inserted, it backs up and removes it. Third, 5' to 3' exonuclease activity. This lets it remove the RNA primer ahead of it and replace it with DNA, a process called nick translation. That combination, building forward, proofreading backwards, and chewing out RNA primase ahead, is what makes it unusual. The Klenow experiment. If you treat DNA polymerase 1 with a protease such as trypsin, it cleaves the enzyme into 2 fragments and 3 activities splits nearly between them. Large C-terminal domain, the Klenow fragment, it retains the 5' to 3' polymerase activity and 3' to 5' proofreading exonuclease activity. This is why the Klenow fragment is the lab reagent even today. It builds and proofreads DNA without destroying primers. Small N termination domain. It carries 5' to 3' exonuclease activity, the primer removal function. Putting it in the context of the fork, on the lagging strand, once an Okazaki fragment is finished, its RNA primer must go. The small domain is 5' to 3' exonuclease removes the RNA primer. The primase actively fills the resulting gaps with DNA. And DNA ligase, here does the final touch, seals the last nick. That's how discontinuous synthesis becomes a continuous strand. Now let's talk about DNA polymerase 3, the holoenzyme. DNA polymerase 3 is the main replicative enzyme and it's not single protein. It's a large multi-subunit holoenzyme. Let's break it down. The core polymerases does the actual synthesis and is made of 3 subunits. Alpha, the polymerases, it builds the new strand. Epsilon, the 3' to 5' proofreading exonuclease which ensures fidelity. Theta, which stabilizes epsilon. The core enzymes are alpha, theta and epsilon, which has 2 copies. So one for leading strand and one for the lagging strand. So both strands are synthesis at the same fork simultaneously. Gamma complex, the clamp loader, which has 2 tau, 1 delta naught, gamma, psi, chi This assembly loads the sliding clamp onto the DNA. Together these adds up onto the roughly 13 subunit holoenzyme. The beta clamp, the secret to speed, the star component of beta clamp, also called the sliding clamp. It is a ring-shaped protein that encircles the DNA and physically tethers the polymerases to the template so it doesn't fall off. Why does it matter? When the sliding clamp is added, it dramatically increases the processivity of DNA polymerase 3. Processivity means how many nucleotides the enzyme can add before it detaches. Without the clamp, the polymerases fall off after just a few bases. With it, polymerase 3 can hold on to and add tens of thousands of nucleotides without letting go. Enough to power through huge stretch of genome at a high speed. The clamp is loaded onto the DNA by gamma complex, which uses ATP to open and close the ring around the strand. Now let's quickly talk about eukaryotic DNA polymerases. DNA polymerases alpha, beta, delta, epsilon and gamma. DNA polymerase alpha. It works with primase to make the initial RNA DNA primer that starts each new strand. DNA polymerase beta. It does DNA repair. Specifically, it does base excision repair. DNA polymerase delta, lagging strand synthesis. DNA polymerase epsilon, leading strand synthesis. DNA polymerase gamma, mitochondrial DNA synthesis. It replicates the DNA inside your mitochondria. Note that neat parallel with prokaryotes, the eukaryotic sliding clamp is called pCNA and clamp loader is RFC. Same logic as beta clamp and gamma complex, but just different names. Let's run the whole fork in order. Helicase unwinds the double helix by breaking hydrogen bonds powered by ATP. Single-stranded binding protein coat the exposed single strands and stop them re-annealing. Topoisomerase or DNA Gyrase relieves the positive supercoiling that piles up ahead of the fork. Primase lays down a shorter RNA primer, giving polymerase a 3' OH to start from. DNA polymerase 3 in prokaryotes extends that primer at high speed, held on by the beta sliding clamp, proofreading as it goes. DNA polymerase 1 removes the RNA primer and fills the gap. DNA ligase seals the final leg. If this breakdown made the replication machinery click for you, that's exactly what this channel is for. And keep learning, keep achieving, and I'll see you in the next video. Thank you.